Yeah, so I guess the question, outside of question is like, what is the function here? Okay, with somatosensory, with the sensory afferent information, you have two different kinds of neurons to think about. Those involved in somatosensory information coming from the environment, and those involved in vision and olfaction, so label here. But with the motor, the efferent information, we can just go ahead and label this is all motor. That's all they're involved in, is motor function. Okay, just plain old motor stuff. That two neuron system that we talked about a little bit ago. Okay, now last section. So again, if you want to just put some dots down here. Okay, to separate the next section. The last section that we're going to talk about are something known as interneurons. I'll put that in brown. Those are interneurons. Okay, and your interneurons are nothing more than connector neurons. So as far as the function, I'll put they're connector neurons, and they're only in the CNS. So they're involved in connecting one neuron to another, and we're going to talk about some connector neurons when we talk about sensory reflexes when we get to the spinal cord portion of the class. And these are just basic connector neurons. There's two different kinds of connector neurons that we need to talk about. The first kind is going to be something known as an anaxonic. So A-N-A-X-O-N-I-C. So that's an anaxonic neuron, just a connector neuron. And based on the name anaxonic, what do you think it doesn't have? Axon. That's it. So it looks like just your basic. We've been depicting cell bodies with dendrites, just like this. That looks like the anaxonic neuron. Only, in addition to the dendrites, one of these dendritic-looking spikes is really an axon. Okay, so it doesn't have the characteristic long axon. It's just a connector neuron. Most of them are dendrites, but one of them is an axon. Don't know which one. Okay, other cells do, but I can't tell. Okay, and then in addition to the anaxonic neuron, another kind of interneuron in the CNS, there's going to be also a multipolar neuron. Just like in the motor system, this multipolar neuron looks just like this one over here, only it has a short axon. So we have our basic dendrites. And the nucleus of the cell body. But this multipolar neuron that functions as a connector has a short axon, followed by the axon terminal. So I'll label that as short axon. And again, this neuron histology is nothing more than what these neurons look like under a microscope. Essentially, structure follows function. All right, the function, whatever the function of those neurons are, they have a very specific function. Whatever the function of the neuron, they have a structure associated with that function. Okay, so somatosensory is going to be pseudounipolar. Vision and olfactory is going to be your bipolar. Motor system, standard multipolar. And your connector neurons, or your interneurons, are going to be your anaxonic, or a multipolar with a characteristically short axon. Questions about that? Okay, histology lesson is over. Let's move on to glial cells. Does anybody know what glial cells are? So essentially your CNS is composed of neurons, and it's also composed of these things called glial cells. And your glial cells, I think the word glial means glue, but they're really support cells toward your neurons. Okay, so your glial cells, and all the next couple of pictures that we're going to draw are going to be glial cells, kinds of glial cells. Now I'm going to focus on it. The first one, the most prevalent type of glial cell in your CNS, is going to be your astrocytes. Oh, by the way, most of these are going to be CNS glial cells. And as we get toward the end, I'll talk about two that are specific to the PNS. But for the most part, these are going to be CNS cells. And the first one that I'm going to talk about is your astrocytes, most prevalent in the CNS, most prevalent glial cell. And astrocytes is going to be a star-shaped cell. So I'll start out talking about these astrocytes. Okay, what I've drawn here at the top is just your basic multipolar neuron. Here's a dendrite, cell body, axon terminal. Okay, so I can label that. That's just your basic neuron. And this is going to be a neuron in the CNS. Okay, we talk about astrocytes and microglial. And down here below, all I've done is drawn my representation of a blood vessel. There's going to be a blood vessel in the CNS. We covered that blood supply the other day. You can pick your favorite, ACA, MCA, PCA, whatever you want to choose. Okay, so this is going to be blood vessel in the CNS. First thing I'm going to do is draw an astrocyte. The astrocyte, like I said, it's a star-shaped cell. I think it's easier to see the functions if I draw little feet off of all the projections off the star. So I'm going to draw a couple little feet. So I'm going to draw a foot here. I'm going to represent my astrocytes in orange. So there's one little foot. And then here's going to be the cell body. And then I'm going to draw another little foot here. Okay, essentially I'm going to make 3 little feet, and all these feet are going to go up and make contact with the cell body of the neuron. I'll label that. So here this is going to be in orange, is going to be my astrocyte. Continue on with the cell, and I'm going to make specific connections of this astrocyte. The feet of this astrocyte are going to come down, and I'm going to make specific connections with this blood vessel here. Okay, and the feet of this astrocyte, when the connections are made together, there's going to be very little space or no space between these feet when they contact this blood vessel. Okay, so they're all tight and close together. Okay. And I'm also going to draw, in addition to the 3 feet attached to the neuron, 3 feet tight together here on the blood vessel. I'm also going to draw 2 feet sticking out in space, or the intracellular space between the neuron and this astrocyte. Just like that. And complete the astrocyte, and the astrocyte is going to have a nucleus. The astrocyte is going to be the most predominant glial cell. Most predominant glial cell is going to be your astrocytes. And the astrocyte has some very specific functions that I'd like to talk about. We're going to talk about a couple of them here. The first one is going to be the tight connections that I've drawn here between these feet of the astrocytes. So if you can see here, I drew them real tight together. Okay. And one of the functions, the first function of the astrocyte that I'm going to talk about here is going to be supporting the integrity of the blood-brain barrier. I'm going to say supporting blood-brain barrier. And by that I mean nothing more than the astrocytes themselves are involved in supporting the blood-brain barrier. Okay, so the blood-brain barrier is very specific, so you don't want everything wandering into the CNS or the area of the CNS. So there's this very specific thing called a blood-brain barrier that prevents unwanted things from entering into the CNS, and astrocytes are involved in supporting or helping to maintain that blood-brain barrier. It's going to be the first function. Question? We should just stretching. Yeah, I know. It's late. The second one is going to be, if you have to think about food, how do the neurons get food from the blood vessel? So the predominant food from the neuron that the neuron's going to want is, does anybody know what neurons prefer? Glucose. Glucose, absolutely. Glucose. Plain old sugar. Okay, brain prefers sugar. Okay, and the second function of the astrocyte is to help bring food or glucose that's free floating in the blood vessel in to the neuronal cell body. Okay, so what I've done here is I just have my food or my glucose in the blood vessel, and the astrocyte specifically is involved in uptaking the food from the blood vessel and transporting it to the neuronal cell body. Got it? Questions? Okay, let's talk about the third function of the astrocyte. We started out talking about that action potential going down, propagating down the axon due to the opening of the sodium channels, but we also talked about the release of potassium. Release of potassium from the neuron into the extracellular space. Okay, so that action potential goes through, you reach a positive point, and then those potassium channels open, potassium floods out of the cell. So what you have is, when the potassium floods out of the cell, you have an increase of potassium in that extracellular space. Okay, so what's going to happen is the astrocytes are going to be involved in the uptake of that extracellular potassium. From action potential. So extra potassium in extracellular space. You don't really want that there, so the astrocyte's going to help keep that environment more neutral. And the fourth, yes? Go ahead.
Uptake of potassium. What?
From the action potential. Yeah, so during the action potential, remember those potassium channels opened and potassium flooded out of the cell to make the cell more negative in charge, membrane potential more negative. So you have extra potassium floating around. So the astrocytes help to take up that extra potassium. Then there's one more function, and it's going to be the release of neurotrophic, neurotrophic factors. Okay, so the astrocyte is going to help to release neurotrophic factors. Has anybody ever heard of neurotrophic factors or know what they would be or what they do? It actually has to do a lot with, well, during development, neurotrophic factors are going to be really important. And also during neural rehab, neurotrophic factors are going to be super important. Neurotrophic factors are going to be released by this astrocyte into the extracellular space, and they're going to help guide axons to where they need to go. Okay, so they're going to help guide axonal growth. You can imagine the patient has had an ischemic stroke, and that part of the axon died. It's no longer working. So that neuron has to make a connection where there used to be one in order to have a person function. And they call neuroplasticity. We're going to learn all about that next year. But the neurotrophic factors actually cause that axon to resprout and make a new connection where it should be with the right therapy. Okay, that's in a nutshell, neurotrophic factors. Questions about that? Pretty straightforward, I hope. And then there's going to be one more thing that we're going to talk about as far as this slide is concerned, and we're going to talk about the microglia. So if I gave you the word micro, what do you think? Small, okay? These are just small cells. These are small glial cells. Great. So your small glial cells, and small glial cells look kind of like this. I'm going to draw them over here. And they have a cell nucleus, and they have little fringes coming off. Just like that. That's going to be your microglial cell. Okay, where the astrocytes had those 4 functions associated with them, the microglia has one job in life, and that is to become the CNS immune system. So microglia, they function as the CNS immune system. And to represent that, I usually like to draw these little structures here. Does anybody know what these would be? Viruses, yeah, they're viral particles. Okay, so viral particles get into the CNS. It's going to be the job of the microglia to seek out and destroy the viral particles. All right, it's just a visual to help with that. Okay, so star-shaped cells, astrocytes, they're going to help in supporting the blood-brain barrier. They're going to help in providing food for the neuron. They're going to help in the uptake of extracellular calcium that's happened after an action potential. And they're going to also promote the release of neurotrophic factors, which help guide axonal growth. Okay, 4 functions of the astrocyte. As far as the microglia, small little cells, their only job is CNS immune system. And I just represented that by it's going to eat up the viral particles, take them out of the CNS. Questions? Let's move on. Moving on. The next one's going to be a little bit of a quick review. Talk about another glial cell, another kind of glial cell that's only in the CNS. It's called an ependymal cell. Okay. Your ependymal cells, here quick review. So if I had to say these are nuclei involved in somatosensory information, relay of somatosensory, does anybody remember what these are called? Yeah, thalamus. One's a thalamus, two thalami. Okay. Then you have your cortical region, so your cortex. There's your midbrain, your pons, and your medulla. Okay. Quick review. Going back and thinking about the ventricular system that we just did, those big hollow spaces that reside in the cortical region and down through the brainstem. Okay, there was one part of the ventricular system that resides between the thalami. Does anybody remember which one that was? Yeah, great. This is going to be your third ventricle. So I'm going to draw my third ventricle in here between my thalami. Awesome. Third ventricles come all the way down here. Okay, and then the third ventricles here. Okay, coming off of the. Oh, let me label that. That's my third ventricle. Okay, coming off at the top sides of the third ventricle, I have a couple little openings that we labeled previously. Does anybody remember what these are?
Foramen of Monroe.
Foramen of Monroe. Good. Okay, this is going to be your foramen of Monroe. It's going to be the opening between the third ventricle, or connection between, or canal between the third ventricle and your lateral ventricles. Remember from the front view, so your lateral ventricles are going to be like horns sticking out here on the side. The big old space filled with CSF. All right, so here's your two lateral ventricles here on the side. Let's label that lateral ventricle. Okay. And my favorite part of the ventricular system is going to be, somebody knows it already. Going to be your cerebral aqueduct, right? That's going to be one of my landmarks here to denote the midbrain. So cerebral aqueduct here connecting the third ventricle through the midbrain. Aqueduct. Okay, and then underneath the cerebral aqueduct, there's going to be another ventricle. Does anybody remember what that one is? There's your fourth ventricle. Good. So coming down here, you're going to make some connectors on the side. Fourth ventricle. And then from the fourth ventricle all the way down through the spinal cord is going to be your central canal. Okay. And that fourth ventricle, let me label that. So again, just a quick review. Here's your fourth ventricle. Okay, that fourth ventricle has a specific foramen that's in the middle. It sticks back toward the cerebellum. Does anybody remember what the middle?
Magendie.
Magendie. This is your foramen of Magendie. Good, Magendie. M for middle, M for Magendie. And then you have these two openings here on the side. Anybody? Luschka, yeah, foramen, good. Luschka, foramen of Luschka. So there's one on either side. Okay, Luschka, L for lateral, two on the outside. Okay, you're wondering why are we looking at glial cells and drawing the ventricular system? That's because the ependymal cells are a kind of columnar cell that lines the inside of the ventricles. Okay, I'll draw that here. Here's going to be my ependymal cells. I'm going to draw little columnar shaped cells. I'm going to leave a space there for a reason. I'll get back to it. But I'm just going to, for the sake of time, I'm just going to draw these. These are your ependymal cells, and I'm just going to draw it inside this lateral ventricle. you should know that these ependymal cells are nothing more than cells that line your ventricles. Okay, and I'm just representing them here as columnar cells. Those are your ependymal cells, and they line all of your ventricles. So for the sake of time, I'm just drawing them on that one lateral ventricle, but you should know that your ependymal cells line all of your ventricles. And these ependymal cells actually have a little hair or a cilia that stick out of them, out of all of them. What do you think the function of the cilia would be for these cells? Any thoughts? What are these cells? What are the ventricles filled with?
To move the CSF.
Yeah, help move the CSF. So they're little hair cells that, with motion, can help promote the flow of CSF throughout the system. Okay. Yes.
Why is there a gap right in front of the
I'm
cells
going to
there?
fill that in it because it's going to be a different kind of cell. Good question. I'm going to draw that here in a minute. Just getting everybody on board. Okay, now, Paul asks, why did I leave a gap? Well, I left a gap so I wouldn't forget to draw in something very important. And the thing that I'm going to draw in, and I'll draw it in as red, is these are a specialized kind of ependymal cell. And I'll draw it in red here for good contrast. I'll just draw 3 of them there. And this special kind of ependymal cell is really just a specialized kind of filter. And this filter is involved in the production of CSF. What was the name of that kind of cell that makes CSF?
Choroid plexus.
Awesome. This is your choroid plexus. Okay, they make your CSF. And you should know that your choroid plexus is nothing more than a specialized ependymal cell. Questions? In a nutshell, that's it. Paul, did you see something?
What's it say under choroid plexus?
Make CSF. Yeah, the choroid plexus is what makes CSF. And the choroid plexus we talked about last week, I guess. And those are nothing more specialized ependymal cells, kind of filtering cells that help make your CSF. That actually make your CSF. Don't help it. You make it. Okay? So kind of linking a little bit what we talked about previously with the glial cells. Moving on to myelinating glial cells. Great. Wonderful. Okay. Talked about all the cells, all the glial cells, and the types of glial cells that I've talked about so far. I said we're in the CNS. Now we're finally going to get to a type of glial cell that's in the PNS. Okay? There's going to be one kind of glial cell or one kind of myelinating glial cell in the CNS and a different kind of cell in the PNS. Okay? We're going to start over here on the left side with the CNS. Does anyone know the name of the myelinating glial cell in the CNS? Oligodendrocyte. Awesome. Yeah, I have a good friend. He used to have a cat named, his name was Oligodendrocyte, but she called him Ollie for short, which I thought was kind of funny. So that's my friend group, right? Oligodendrocyte. Okay. Oligodendrocyte is going to be the name of the myelinating glial cell in the CNS. Okay. And we're going to start over here. This is going to be your basic multipolar neuron. Okay, with dendrites, cell bodies, nucleus. Here's the axon terminal. Okay, we've covered that hopefully well enough today. And the myelinating glial cells, I'm going to represent that it's wrapping around this axon by this little swirl here. So this is just a little swirl that's wrapping around the axon. And the cell, turns out that in the PNS, the oligodendrocyte itself can wrap not only multiple areas of the same axon, but axons that are residing close in three-dimensional space for multiple neurons. Okay, so this is the myelin sheath. This little swirl here is just the myelin sheath that the oligodendrocyte produces. And there's some feet coming out. And this particular oligodendrocyte, representing here, is responsible for wrapping myelin around 3 different areas of the same axon. Okay, so this guy is your oligodendrocyte, and this is your myelin sheath. M-Y-E-L-I-N sheath. Okay. So these oligodendrocytes essentially send out appendages. The appendages wrap around the axon to create a myelin sheath. Okay, that's CNS. Moving over to PNS. PNS is similar, just a little bit different. The PNS has a specific kind of myelinating glial cell. Does anybody know the name of that one? It's going to be your Schwann cell. S-C-H-W-A-N-N. That's a Schwann cell. Okay, oligodendrocytes are in the CNS exclusively. Schwann cells are in the PNS exclusively. I had to represent the Schwann cell, similar to the oligodendrocyte, only just a little bit different. Each individual cell, each individual Schwann cell, is responsible for myelinating just a small area of an axon. So that is representative of just one Schwann cell. So here's your Schwann cell. So here, one oligodendrocyte could wrap 3 different areas of the axon. To wrap 3 different areas of this axon in the PNS, you actually need 3 different Schwann cells. Everybody see that? So everybody see the difference? Okay, individual Schwann cells just wrap a small area of an axon, creating a myelin sheath around that axon. Whereas in the CNS, oligodendrocytes, one oligodendrocyte can wrap multiple areas of an axon. It can also wrap myelin around neighboring axons from different neurons. So oligodendrocytes, much more efficient. One more cell type that I'd like to talk about, and that's going to be in the PNS. Pretty basic cell. Okay, this is going to be something in the PNS known as a satellite cell. S-A-T-E-L, satellite cell. And your satellite cell in the PNS is a kind of glial cell that is basically a neuronal support cell. N-E, support cell. Okay. Kind of like the astrocyte, does whatever's needed. Well, the satellite cell, kind of like that for the PNS neurons. Questions about that? Anybody? Does that make sense? Yes? Yes, Ben?
The myelin sheath, is that all 3 tubes that are on that axon, or is it just one of them?
PNS or the CNS?
In the CNS.
Well, all of them are myelin sheaths. So everywhere that I've indicated, so all of these guys, if that's not clear, all these here, all these are all myelin sheaths, and all of these over here are myelin sheaths as well. Okay, the myelin sheath is what's wrapped around. It happens that the myelin sheath is created by the entire Schwann cell here, where okay.
Thank you. That clarifies.
Okay. Anybody else? Yeah, Jonathan?
Can you explain why you said that the oligodendrocyte is more efficient than the Schwann cells?
Because the oligodendrocyte, one oligodendrocyte can wrap around multiple areas of one neuron or multiple areas of multiple neurons.
But the Schwann are all separate.
These are all just individual cells. So it takes one oligodendrocyte to wrap one axon. Here it takes 3 Schwann cells to wrap one axon to cover the entirety. How it is? No, not a stem cell. The neural so it's a glial cell, so it's supportive in nature. You know, I said the astrocyte has lots of different functions. So your satellite cell might perform some of the functions that an astrocyte would see. So basically, whatever feeding, all that stuff can be performed by satellite. Any kind of supportive function. You should know that's the satellite cell. Just a different kind of glial cell. 2 different kinds of glial cells that we saw in the PNS are going to be the Schwann cell and the satellite cell. All the other cells we talked about are going to be specific for the CNS. Any other questions before we move on? Okay, hopefully that was pretty clear. This is a lot of just basics. I mean, things that you need to know neuroscience-wise. We'll move on to the last drawing for the day. Yay. This is going to be neuron communication with and without myelin. Okay, neuron communication with and without myelin is going to be the last one. And we talked about, and just to make it easier, here's 2 neurons in sequence. Okay, we've seen this is going to be your dendrites in your cell body, your axon terminal. Here's your next neuron in sequence. Okay, so this is going to be your synaptic cleft right here. So presynaptic neuron, postsynaptic neuron. Okay, we're just going to start right there. We're not going to worry about the PNS and the CNS on the bottom. We'll get there in a minute, but we're going to start up top. Okay, so in thinking about drawing the oligodendrocytes versus the Schwann cells, I think that the Schwann cells are a little bit easier to draw, so I'm going to choose to myelinate this neuron, wherever it is, just with Schwann cells for simplicity. Okay, so this is going to be my Schwann cell here. If it's a Schwann cell, it exists in the PNS. But okay, there's one Schwann cell there. There's another Schwann cell here. Okay, there's another Schwann cell. So essentially, all that axon is wrapped with 3 different Schwann cells. I talked about the transmission. So these are the dendrites, and they contain the mechanically gated channels, and mechanically gated channels associated with the environment. So if you think about pain, for example, they're going to, in response to a pinprick, they're going to cause the opening of those mechanically gated channels, causing sodium to rush into the cells through the channels. Okay, so let's say that we have sodium coming into the cell, okay, in response to some sort of sensory modality, and sodium comes into the cell, and then we reach the minus 50 millivolts, and that's transmitted through the axon hillock, which results in an action potential going down the rest of the neuron. We spent the first part talking about this today. Okay, so to represent this action potential, I'm just going to put here, the positives are going to be action potential. Okay. So does anyone know anything about the importance of myelin? I mean, why is myelin so important? Go ahead. Yeah, it speeds up the rate of the action potential propagation down the axon. And it does that because it sort of skips over the areas of myelin. So you have action potential here, skips over this, but then it appears here. So it can skip that whole region because it's coated with myelin, the fatty sheath, and appears here. Okay, skips here. So you have an increase in the speed of propagation of that action potential down the axon. Okay, and that's going to be the important part of myelination. As I said, there's this process has a name. The jumping of the action potential basically over the myelin sheath has a name. Does anyone know what that name is? It's called saltatory conduction. S-A-L-T-A-T-O-R-Y conduction. Okay, saltatory conduction. And that means nothing more than the action potential jumping over the myelin sheath. Okay, and appearing in areas where that myelin sheath isn't present. Thinking about areas of the axon where the myelin sheath isn't present, that has a specific name also. Does anyone know what so this area here and this area here? What does it, you know what that? Yeah, do. Go ahead. Nodes of Ranvier, yes. So these are called nodes of Ranvier. Okay, and nodes of Ranvier are areas on the axon that aren't covered with myelin. Okay, and that's where the action potential jumps from node of Ranvier to node of Ranvier, just jumps right over the myelin sheath. Questions about that? That makes sense? Is that something you all have seen before or heard about a little bit, maybe? Sort of? Yeah, some people. Okay. All right. Now, let's go ahead and go down here to where I have CNS and PNS listed. Okay. On the CNS side, we're going to start over here. And for CNS, does anyone know of any demyelination disorders that affect the central nervous system? Here. What's the main one? What's going to affect the oligodendrocytes and cause demyelination in the central nervous system? Go ahead. MS, yes. Multiple sclerosis. Perfect. In the CNS, there's going to be a disease process known as multiple sclerosis, or MS. Okay, things that you need to know about MS. MS is an autoimmune disorder. Okay, it's due to a demyelination in the CNS, which means that the oligodendrocytes are affected. Right? Because the myelinating cells in the CNS are going to be your oligodendrocytes. Okay. Another thing that's important to know, we'll talk a whole lot more about multiple sclerosis next year in neuro rehab. But for now, it's going to be important to note that with the disease process of multiple sclerosis, signs and symptoms are going to be very variable between patients. And because of this, it often takes a very long time to be diagnosed with multiple sclerosis. Lots of different kinds of multiple sclerosis. Probably the most common and the first form that usually appears is known as relapsing remitting, where you have this problem, let's say visual disturbance. Okay, you have a visual disturbance, and I mean suddenly you're having trouble seeing over here for whatever reason, and it's there for a while, and then it just goes away. That's relapsing remitting. Okay, and then maybe in 6 months or so, you might have some numbness and tingling in your hand. And it's kind of annoying, and it's just there, but then a couple months later, it just goes away. So that's going to be the relapsing remitting process of multiple sclerosis. And because of this, it's often difficult to get diagnosed. We'll talk about diagnosis next year. That makes sense? Moving on. So this is the CNS. And again, to separate the PNS and the CNS, let's go ahead and put some dots down there. And there's a couple of disease processes that involve demyelination in the PNS. Does anybody know the main one that's an autoimmune problem?
Is that GBS?
Guillain-Barré syndrome. Awesome. Yes, this is going to be Guillain-Barré. Guillain-Barré syndrome. I'll abbreviate GBS. Okay, again, we'll talk more about GBS next year. But you should know it is, again, an autoimmune disorder. Okay, GBS, autoimmune disorder, and this time the autoimmune disorder, it's going to affect your Schwann cells. It's a demyelination. This time the demyelination is specifically in the PNS, right? And affects your Schwann cells because those are the myelinating cells in the PNS. Whereas, and one thing for multiple sclerosis, one thing I neglected to put here, this is a lifelong problem. Once you're diagnosed with multiple sclerosis, it is a lifelong problem. Where Guillain-Barré, approximately 80% of patients diagnosed with Guillain-Barré have a full recovery in 2 years. Okay, it's a little bit different than multiple sclerosis. Okay. What happens to the other 20%? I don't know if anybody's thinking about that poor 20%, but they don't get better. They might get stuck in one particular place and be left with all kinds of deficits. Okay. Happens. And then there's one more. This one, so multiple sclerosis, Guillain-Barré, both autoimmune disorders. The last one that we're going to talk about is called Charcot, C-H-A-R-C-O-T, Marie, M-A-R-I-E, tooth, T-O-O-T-H, syndrome. In contrast to the other two that are autoimmune disorders, Charcot-Marie tooth is genetic. So you're born with it. Essentially, Charcot-Marie tooth is there's a specific protein. It's called connexin that it resides inside the Schwann cells. It's important for myelination, and that's a problem with the connexin in that protein. Too much information. Don't just know it's genetic. It's good. Signs and symptoms are going to be specifically decreased strength in lower extremities, decreased strength and decreased sensation, specifically in lower extremities. Okay, a lot of information today. Talked all about neurons and action potentials and neuronal histology, all the glial cells, and a couple of different problems with myelinating glial cells.
What is that box? So strength, lower extremities, and what's the bottom one?
Decreased sensation of the lower extremity. Yeah. Decreased strength and decreased sensation due to the demyelination. And again, you're born with it, but symptoms don't usually appear soon after birth. It takes a while to acquire the decreased strength and sensation. And it might be a patient population you work with in your future if you work in neuro. Okay? All right, let's go ahead and take a 10-minute break, and then what I'd like